All right, we've got a great show set up for you. I've got John Vandermosten. He is the biotech engineer and analyst for Small Cap Research, or Zach Small Cap Research, and he's got a guest today, Steve Hoffman. He's going to bring him on in just a minute. We're going to talk about the non-toxic cancer treatments and the multiple types of cancer that they work with. John, welcome to the show. Thanks for being in studio with us.
Great.
Yeah.
Thank you, Michael.
Great.
And as you said, today we have the CEO of Tyme Technologies, Steve Hoffman. And Steve founded the company along with his COO, Michael Demerjian, back in 2008. And they now have a phase 2 asset for recurrent non-metastatic prostate cancer called SM88. Steve, welcome to CEO Money, and how are you doing today?
Very good. Pleasure to join you. Thank you.
So you had a career as a physicist prior to your biotech days. And can you share with us about that a little bit and give us more background on yourself and Tyme Technologies?
Basically, my story is, I guess, interesting. I started as physics and electro-optics. I dropped out of college a little before my graduation to work in the polymers improvement for Bausch & Lomb at the time, and that was their Ray-Ban division. And then I went from there to be a gun for hire, I would call it, for just about, probably a dozen other companies and a dozen other processes that needed problem solving, including things as significant as the VAPE program, which is working on aerospace for NASA, Joint Strike Fighter, the F-35, and then much more pedestrian things like, for example, biomedical joint implants and pyrolytic carbon heart valves, and I guess right down to things like saw blades and automotive processes. So my background is essentially one of doing a lot of different things. The company came to pass, ironically, because I was helping out with the creation of one of the systems for proton therapy and realized that in proton therapy, we were using free radicals to kill cancer. And I realized kind of in that moment that there must have been a major misconception about cancer. Here's something that we believe cancer was caused by free radicals, and suddenly we're finding out we can kill them with free radicals. So I started to take apart cancer as a conceptual piece, and when I looked at it from a different perspective, there became obvious opportunities to treat it differently or to potentially remediate it differently. And so Michael Demerjian and myself got a little courageous and formed, a company and ended up treating an individual and then another. And each time we treated people, we found that we saw benefit. So we then went into a much more formalized mode on domestic soil and really came up with what is now known as Tyme Technologies.
Well, thank you for sharing that background. And, we've talked about on this show a few other different approaches to cancer. CAR-T is one, checkpoint therapies, checkpoint inhibitors is another. How do you fit into the paradigm, I guess, of cancer research with your product?
Well, again, what I did, which was a little bit different, was I said perhaps if cancer is so pervasive, it isn't necessarily an invasive thing, but rather it might actually be a natural function of the immune system gone rogue as opposed to being just an opportunistic pathogen that created a new problem. So when I started to look at cancer, I said, let me look at where it regulates in consistent ways with the body. In other words, we have cancer across a lot of spectrums of things. We have cancers caused by a lot of things, but the cancer itself is cancer. In other words, why do they call it that? What is its commonalities? And it turned out that its commonality, at least its greatest, most robust truth, was that this very fragile, very weak cell was protected by something called mucin. So various different technologies have looked at how to either strip away that mucin from its essential cloak of invisibility, which is some immunological processes, or how to modify, or even how to topically poison. What I said was, how is this mucin actually manufactured by the cell, and how can I potentially stop that from happening? So I looked at a metabolic approach. It turned out cancer follows the human body in certain processes, case in point, becoming ketotic or ketogenic. In other words, when the body thinks it doesn't have enough insulin, or the body thinks it doesn't have enough sugar, it goes into a ketotic state basically designed to save our life so our core organs can get what's necessary. So I looked at cancer, and surprisingly enough, when the body does that, the cancer goes into the same conservative state. In other words, it goes into a state that's trying to support the survival of the host. So how does it survive then? Because that defiles its capacity to have a diet of glucose. And as it turns out, the next thing it consumes is amino acids. So I found a way to denature. By denature, what I mean is sort of the same way sugar substitute is like sugar but isn't sugar. Found a way to denature one of the essential amino acids, which was tyrosine. And by producing this and getting the cancer to consume it, the cancer metabolically becomes weak. What that means is the cancer can't make the proteins that are essential for this mucin manufacture. And when it doesn't have mucin, it's literally like a turtle without its shell. It is now extraordinarily vulnerable, and other processes can kill it. And so that was really the idea. We call ourselves a metabolic approach, but in so many ways we're synergistic with the other approaches.
And why is this work so well in prostate and pancreatic cancers? And, you know, just for our audience, prostate is one of the most common cancers out there, and pancreatic is a little less common, but one of the most deadly. How does SM88 particularly work well in those specific types of cancer?
I think what you're asking is a terrific, almost more generalized question than that, which is how does SM88 work in cancer, and why does it work so well specifically in any one cancer? Ironically, it's not really better in any one cancer than any other cancer. When we put our denatured tyrosine into the human body, it really is functionless to the rest of the cells in the body. Those cells can manufacture their own tyrosine out of phenylalanine. So the only cells that are devoid or missing this tyrosine product are the cancer cells, regardless of where they're located. And they're voracious at this point. The voracious appetite of cancer is saying, I've got to produce ATP, I've got to produce energy. So we put it into the system, and it literally gets to 100% of the body. And the way we know that this works is there's a fairly similar thought in one of the imaging agents. When a person goes in for certain scans, they use imaging agents to accentuate, particularly PET scan agents, to accentuate, the idea if you've got SUV, which is uptake or metabolic excitement of these cells. So in the same way that an imaging agent seems to magically get to the disease in any place it is, our denatured tyrosine and its combination of other materials, which we call SM88, does the same thing. So in prostate cancer, it's particularly visible to us because prostate cancer produces two things that we can measure external of the actual disease. One we all know, which is PSA, prostate-specific antigen. That's basically the cancer's, production of antigens, which we're able to measure in the bloodstream. And the other is something called CTC, which is circulating tumor cells. We can measure both of those. I'm going to call them chemicals because we don't have to biopsy a person to do it. We can measure both of those things, and we can measure our effect on that. And if you look at our clinical data, we not only suppress the PSA increase, but we actually stop the circulating tumor cells, meaning we stop something that may turn out to be a powerful precursor to metastatic disease. So if you can keep prostate cancer localized, it's a much more treatable thing, and our medicine ultimately will do that, but it's treatable by other mechanisms as well, including surgery, including other, radiation options and things that can be done. So prostate cancer works wonderfully.
So it sounds like
pancreatic cancer, let me just be very honest with you. When you do a clinical trial, you have what are called endpoints, and hopefully those endpoints are things like PSAs or things like CTCs. Hopefully you've got a viable person, and the endpoint of that trial is a chemical or something you can measure. With pancreatic patients, the endpoint is survival. The endpoint is binary. These poor people don't have long lives. So we got into pancreatic because we did preliminary work on it with exceptional results. Our goal there is simply to lengthen survival, hopefully good quality of life, but lengthen survival as an absolute, as I said, binary goal. In that cancer, we think we're going to have a fantastic result.
Steve, I hate to interrupt, but we've got to go to a commercial break. We're going to continue the conversation right after we get back, all right?
Certainly. Thank you so much.
All right. You're welcome. All right. We've been listening to John Vandermosten of Zacks Small Cap Research, speaking with Steve Hoffman. He is the CEO of Tyme, Tyme Technologies, TYMEINC.com, and their symbol is TYME. Believe me, I'm got to look at the price. You're very proud to bring this content out at this share price. You got to take a look at it. We'll be right back. You've been listening to CEO Money with Michael Yorba. We'll be right back after these messages. Welcome back to CEO Money. I'm Michael Yorba, your host. All right, we're talking with John Vandermosten, who is sitting in and hosting the biotech segment for Zacks Small Cap Research, and he's speaking with Steve Hoffman, CEO of Tyme Technologies, symbol TYME. And John, welcome back to the show.
All right, thank you.
Steve, welcome back to the show.
Thank you.
You guys were having a brilliant conversation about how you're going about curing cancer, the many forms of cancer. Hopefully I can say it that way because I don't want to get in trouble with the the authorities on that. But please, John, please pick up where you're going. I was riveted.
Let me pick it up a little bit. So, Steve, one thing I wanted to ask you was about just the safety profile of the drug. You mentioned that there's a dysfunctional tyrosine derivative, or maybe I read that in your report. And how it takes away the layer of the cancer cell. Now, how does it, or how do you prevent this from having negative effects in other part of the body?
Well, the interesting thing I've done in this particular combination is I looked at some drugs that already had pretty well understood histories. And, for example, we create this ketotic or ketogenic state by using something called a leucine aminopeptidase inhibitor. And that sounds all very complicated, but at the end of the day, the chemical that's being used in this is well understood, just as the chemical that helps triglycerides and oxygen be available to the cancer and the chemical that helps induce that catalyzed relationship. All of those are well understood. And the amazing thing is that the one chemical that we innovate with, the one that's our new created molecule, which is the tyrosine, as you said, in the nature tyrosine, this particular chemical is only viable. In other words, it's only consumed by the desired cell, in this case, the cancer cell. It has no value to the rest of the cells of the body, as has been well proven, as I said, in similar kinds of experiments with imaging. So it's essentially, I hate this term because it's not a correct term of science, but let's use it differently. It's minimally toxic. We haven't so far seen toxicity. I mean, we've seen tiny little things where people feel tired for a couple of hours. We've seen things where people have a little bit of hyperpigmentation. It's called, where you see a little bit more melanin being produced, but nothing that's radical. But beyond that, they're just, actually what I want to say is something a little different. Whereas most people get cancer, tragically, the mechanisms or the methods that are used to treat them make them feel terrible. Our patients have exactly the opposite. Our doctors have been, testimonies have stated that the patients look and feel better within a couple of days of starting, and typically within 14 days, there's an evaluable, you can evaluate that there has been some remedy, that there has been an improvement in the cancer, certainly an improvement in attitude, certainly an improvement in general function. So I dare say we're not only minimally toxic, but in a great many cases, we're providing benefit to the patient where we actually see an improved person, an improved personality.
Okay, well, thank you. And I think one of the terms that you use is actually it is a non-toxic cancer treatment. Can you expand on that a little bit?
Yeah, when you think about most chemotherapies or most therapies, they're dealing with a lot of cells. You're getting what you want to have happen and getting what you don't want to have happen. And a lot of alkalizing agents, in other words, a lot of, let's say even platinum drugs, because people have heard of those. What happens is they create discontinuity in the cell that you desire this, in the cancer cell, but they also do that across the rest of the body. They have a systemic profile of complications. Sometimes they're severe. Hopefully they're not. But people know a lot of the side effects. For example, when you involve yourself in rapidly dividing cells, you could see the situation where people have hair loss, things like that. We just do not have that effect in our patients. There was a wonderful article that People Magazine Online did on Christian Hodge, who's one of the patients we've treated. She was on a television show, and beautiful then, beautiful now. In other words, we see that our patients do not deteriorate. So when I say minimally toxic, what I'm suggesting is that the cancer cell is affected by this. The rest of the body is not adversely affected by the medicines. There is no problem that we create that a person will suffer, at least none that we have seen so far. And there are criteria, this is Reese's criteria, there are things that are extremely specific and extremely regimented about this subject. And when you use all of those criteria, all of them, we show a demonstrable benefit to these patients.
Steve, it's Michael Yorba. I just want to jump in and ask you a quick question for me. Are there any other treatments that go along with the treatments that you're working with to treat your patients? In other words, radiation, chemo, blah, 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 the other types of classic cancer treatments, or does this take the place of all of it?
You know, that's a terrific question, and let me start by saying that probably the hardest job in this world is being an oncologist, because unfortunately such a high percentage of the people you're dealing with are so very sick. Mm-hmm. And oncology has become something of an art in addition to being a science. And I don't say that in some way to disparage it. I mean it very seriously, that sometimes what an oncologist will do is they will see a patient having had, resolved their disease, which thank God happens, and they say, let's go one more round of therapy just to sort of be a precursor, to sort of prophylactically try to ward off that problem again. There will always be collaborations between our technology and other technologies as a standalone, which is how we do our clinical trials. Our product has shown demonstrable benefit, complete response in large amounts of patients, extremely extended lifespans, even on stage 4 patients, even on the sickest patients where there is an expectation that their life will be, somewhat limited. We have seen dramatic results, even complete responses, even in pancreatic patients.
And Steve, I wanted to ask a little bit more.
When the oncologist sees it, I want them to use our drug with other drugs. I want them to be comfortable. And the nice thing is, if you don't do damage, you've created a very clean canvas to let people try other things with it.
And Steve, just on the combination therapy side of things, one of the more popular combination therapy drugs are checkpoint inhibitors. Do you think there's any potential synergies that you could achieve here?
At this point, I can't tell you from a standpoint of having had done science to give you the straight answer, but I'd be very excited to see those work together. I would be honestly very excited to see a person who attempted to use those to the benefit of the patient.
Okay. And, there was one technical question I wanted to ask, and you did touch on this a little bit earlier, but related to the Warburg effect. Can you explain what this is and how it facilitates efficacy in your drug?
Okay. The basic idea is that cancer dies with too much oxygen. Cancer is not good at using oxygen. And when you can overload oxygen into a cancer cell, you can kill the cancer cell. What we do in this case is we say, when somebody says overload oxygen, that sounds like a wonderful thing. That sounds like breathing. But sometimes oxygen is oxidation, as in rusting metals, as in materials that are, in some way met by an acidic condition. So the body has the ability, internally and externally, and we know this, to create acids, to create what amounts to free radicals or available oxygen that's waiting to react. What happens with the cancer cell is that the cancer cell has this mucin layer, has this protective layer that allows this very fragile cell to exist in what amounts to a microenvironment that's noxious, that would kill it but for this one, sort of membrane. And what we're able to do is to limit the amount of that mucin, limit the amount of that protection. So all of a sudden, this simple material like oxygen can get in through the form of mild acids, can get into the cancer. We use triglycerides mostly, which people have certainly heard of, but those form around the cancer. And by potentiating the reaction, by catalyzing that reaction, by giving these electrons a way to move into the cancer, they carry the oxygen with it, and that oxygen actually will kill the cancer. In other words, at the end of the day, cancer is not a monster. It works out to be a monster because of where it is and what it does, but cancer is actually a frail little thing that is built up by almost armor plating.
And Steve, we got about 20 seconds left. Are there any last words you want to share with us? And we want to mention, again, your ticker symbol is ticker TYME. Any last words for the last 10 seconds here?
I ask everybody to take hope away from this and to please look at our website, www.tymeinc.com, and know we're out there and we will someday be contributing, we hope, to all your family's health.
All right,
in a good way.
Thank you, Steve. Great to have you.
All right, you've been listening to Steve Hoffman, CEO, Tyme Technologies, TYMEINC.com, symbol TYME. And by the way, just as a little footnote here, they have the Phase 2 clinical trials also here in Dallas-Fort Worth area. All right, you've been listening to CEO Money with Michael Yorba, John Vandermosten of Zacks Small Cap Research. We're going to take a brief break, and we'll be right back on the other side of this break with Judge Graham.